I'll just take a few minutes and introduce all our panel members, and then we'll get into the panel presentations and discussions. To begin off, we have Professor Immaculata De Vivo, who has over 280 peer-reviewed publications. De Vivo is a professor of medicine at Harvard Medical School, Brigham and Women's Hospital, and professor of epidemiology at Harvard T.H. Chan School of Public Health. She's also an international leader in molecular and genetic epidemiology of cancer, and her research primarily focuses on how genetic variants interact with the environment to influence susceptibility of hormonal cancers, especially endometrial cancer. Her studies have received worldwide recognition, notably by NASA, with whom she collaborated to study the effect of long-term space travel on telomere length using blood samples from monozygotic twin astronauts. So together with Daniel Lumara, De Vivo has written 2 award-winning books, both of which were bestsellers in Italy. So you can definitely see the range of expertise she brings to the conversation. Next, we have Dr. Lerato Majara, who holds a PhD in human genetics from the University of Cape Town. Dr. Majara has investigated the genetic risk factors associated with schizophrenia in the South African Xhosa population and is currently a senior fellow in the Global Initiative of Neuropsychiatric Genetics Education and Research, better known as GINGER, program at Harvard T.H. Chan School of Public Health, and is currently pursuing a postdoctoral fellowship on the neuropsychiatric genetics in African populations. And I'm sure we'll be seeing later on as to how scanty the research within the region is, and hence why we're having this conversation. Finally, we'll have our own Professor Zul Merali, who is a founding director of the Brain and Mind Institute at the Aga Khan University. He's a neuroscientist and a pharmacologist by training from McGill University, University of Ottawa, and Wharton, with over 40 years of academic engagement, and has published over 250 peer-reviewed scientific publications. He has used his proven academic and entrepreneurial ability to add value to forward-thinking organizations in addressing the mental health problem locally, nationally, and globally. And as you've seen through his presentation, the objectives are to build and or to re-engineer organizations to deliver outstanding results and motivating and encouraging others towards positive change. Hence the Brain and Mind Institute. Without further ado, we'd like to invite Professor De Vivo to take over and share her keynote. Thank you.
Thank you, Kendi. And before I share my slides, I'd like to wish everyone a good day, a good evening, good afternoon. It is morning here in Boston. I'd like to thank Dr. Morelli and Dr. Mahara for being my co-panelists today. It is an honor for me to be part of this conversation today. Okay, so today I'd like to share some of the work that we've done in my lab with collaborators on the impact of epigenetics and mental health. So epigenetics, just briefly for everyone to understand, is changes that happen to the genome, but above the genome, so without changing the actual sequence of the DNA. And one of the markers I like to call that I work with extensively, at least since 2007, has been with the telomere biological markers. So telomeres are the ends of your chromosome that protect the inside of your chromosome. As you could see there, they look like shoelaces. And in the way a shoelace protects your shoelaces from coming apart, telomeres protect your chromosomes from coming apart. They're there to protect the inside of the chromosome that has all the genes, the regulatory regions. It maintains the fidelity, the robustness of the information in your genome during replication, and it's very important for genomic stability. So my initial, these are repetitive sequences shown there. There are these letter repeats, CT8, GGG, that run over thousands of base pairs. And telomere themselves, the lengths are maintained by an enzyme, telomerase, for which was awarded the Nobel Prize years ago by colleagues Elizabeth Blackburn, her postdoc at the time, and others, Greeter, Professor Greeter, and Jack Sostak. So this telomerase enzyme is critical for maintaining the length of telomeres, but I will remind you that it's downregulated or turned off very early on in life, about 15 weeks of gestation in utero. And so as a consequence, stem cells and germ cells have upregulated telomerase, meaning that the telomere lengths are remain maintained. But in somatic cells, such as a liver cell, a cancer skin cell, the telomere themselves shorten over time as a normal process. So what we're interested in is accelerated shortening above and beyond what normally shortens as a consequence of DNA replication, oxidative stress, and inflammation. In this illustration, I show that telomere length over time. You can see that the first 20 years of life, you lose the most of your telomeres, approximately 2 kb, but you continue to lose throughout midlife until the end. The point of this illustration is that telomere loss in somatic cells where you don't have telomerase is most profound early on. It is, and a telomere length is pretty much established at birth, but again, you lose over time. And this is why it's often been referred to as a marker for aging, a biological marker, or the clock. And my initial interest in telomere was when my early days at Stanford was as a molecular mechanism for cancer. But it turns out that measuring telomeres goes well beyond cancer. And I will continue to talk about just some of the dynamics we know about telomere length, because it helps understand the context of the talk, which is that we're born with a certain length, and it's very variable because telomere length is determined not by your genetics as well as the environment. And that's why it's an incredibly important biomarker, objective marker to study not only cancers that are not that are a consequence of gene environment, but also in this particular context today. So they are variable and they shorten over life, as you could see here, by half, if we can all reach to 100, and they shorten as a consequence of normal DNA replication, oxidative stress, and inflammation. Okay, so there's a normal loss, about 25 to 50 base pairs per year. But what we're interested in, again, from a public health standpoint and from a research standpoint, is accelerated loss. Those individuals that lose more than they should normally lose. And so this biological clock has become very instrumental in studying almost all diseases, as well as just exposures, because it's an objective measure that's usually a supplement to questionnaires. So again, to understand telomeres, females have longer telomeres than men, and this is determined at birth. There's also differences by ethnicity, and I've provided some of the papers that I've worked on. One of the more interesting papers that I worked on with my colleague Avi Abraham and others is that we had samples from hunters and gatherers where we saw that Africans have longer telomeres compared to even African Americans as well as Europeans. So there are interesting facts that we know about telomere length that we incorporate in our studies to let us understand impacts of confounding, or to remove factors that might interfere with objective findings, to have an objective understanding of our findings. So I want to just briefly go through this. So the idea is that we measure telomeres through blood samples or saliva, but to show you here in this paper, again, by colleagues, is that the blood sample or the cheek sample, where we take your DNA, is a very good proxy for what's going on in other tissues of your body. Within an individual, the correlation is very high. Well, with respect here, there's muscle, fat, and skin. Across individuals, there will be a lot of variation because, as I said, telomere measurements are a product of your genetics as well as your lifestyle or environment. I like to say how much life one has lived. So you can take two 30-year-olds or 40-year-old females, right? So we're adjusting for age and gender, but they can have completely different telomere lengths because it is a measure of who their lifestyle. Are they obese? Do they smoke? These all have impact on your telomere length. So we know that telomere is associated with telomere length is associated with mortality. In this very large study done by our Danish collaborators, you could see that mortality is pretty well captured by telomere length. Short telomeres predict mortality. We also know that there is a trade-off, like a lot of other things in biology, the trade-off of malaria and sickle cell anemia. In this case, the cancer CVD trade-off is pretty well established by this point. The idea is that short telomeres predispose you to atherosclerosis, but long telomeres predispose you to cancer. And the details for that we don't have time to go into today, but to show you, to emphasize that greater resistance to cancer comes at a cost of greater susceptibility to cardiovascular disease. So we're all looking for that Goldilocks level. And here is a paper that we were part of, which illustrates the cancer CVD trade-off at using Mendelian randomization technique. And you can see that longer telomeres at the which is represented at the top in cancers like glioma and ovarian cancer predispose you to them, or as you come down to shorter telomeres, you see they predispose you to cardiovascular disease. Again, just some facts about basic facts about telomere length. But to summarize, what we know so far is that short telomeres are associated with, of course, a decreased life expectancy as a result of DNA damage, increased rates of age-related chronic diseases such as CVD and cancer, but again, within the context of the trade-off. And we know that the loss is accelerated by oxidative stress and inflammation. And so that leads me to stress. And I have become intimately associated with stress through work with collaborators since about the year of 2009 or 2010. And the idea of psychosocial stress here is predicated on the fight or flight pathway, which was brilliantly summarized by a big fan of mine, which was Robert Sapolsky last week. It was a joy to listen to him, but I wanted to just briefly introduce it very briefly so that the context of the remaining slides will make sense. So Hans Seeley, the father of stress, it's not the stress that kills us, it's our reaction to it. And the consequences of chronic stress, this term has been used very frequently. Stress gets under our skin, and we know anecdotally when we are stressed, we don't look really great, and we're often told stress is aging. And so the anecdotal anecdotal stories are finally being shored up by science. As I like to say, the science is catching up. And so exposure to stress and adversity across the lifespan is associated with increased morbidity and mortality, which I've already shown. And that is stress and adversity have a lasting impact on general physiological processes. And so allostatic load, again, briefly, was covered very well by Professor Sapolsky. And I had the privilege of having conversations with Bruce McEwen at Rockefeller University, the great professor who was the mentor to Professor Sapolsky, who says that we know that environmental complexity changes the brain and that it comes to haunt us in terms of socioeconomic status, poverty, and things of that sort. He also stressed that the chronic stress disproportionately affects marginalized people and increase their risk of illness. We know this very well by many examples. So until recently, there are a few candidate markers of this accumulation of physiologic damage. And therefore, I propose telomeres as an objective marker to assess psychological stress, environmental stress, physical stress, and because it has a genetic component, also to understand the genetics. So one of the very first studies that showed the impact of stress on our DNA was done by Nobel laureate Elizabeth Blackburn, who showed in her PNAS paper years ago that telomere shortening was accelerated in women who took care of children with special needs. What was even more interesting in that paper was that it was not just the stress, but women who had perceived stress suffered disproportionately from telomere loss. This was a talk that I was completely inspired by that she gave at Harvard years ago, and I became very much enamored by this concept, because we know, again, within my studies of cancer, that there are many components, multifactorial reasons, risk factors for cancer origins. So in my lab at the School of Public Health, we measured telomeres, and we've to date measured about 100,000 in collaboration with many people. And one of the studies that came out of my lab in collaboration with Professor Drury at Tulane was the impact of the early childhood adversity as measured in Romanian orphans. This study was an eye-opener for me, and I have provided the papers for people to read, but essentially what was very interesting about this work that showed that children who are suffered horrific early events in childhood have shortened telomeres very early on. But my papers can be picked up by local news in the science section. What was remarkable about this paper was that it was picked up in the New York Times, not by the science section, but by the editorial board. And that made it very obvious to me that the science is now having social implications at the social level. That inspired me to continue these studies. And again, I show you some of the papers where we looked at the impacts of psychosocial stress, perceived stress on telomere length. We also, in collaboration with Olivia Okereke, looked at the impact of phobic anxiety. And I will tell you, even though I'm trained as a cancer researcher, I've learned so much at the altar of these brilliant psychologists and psychiatrists who studied these diseases. And one of the reasons we were so fortunate to see an impact of phobic anxiety on telomere length, and here is the graph to show that the more anxious, the more phobic anxiety someone had as measured by the Crown-CRISP questionnaire, the shorter their telomeres. So here again, we have a questionnaire assessment shored up by an objective biological marker that accumulates the impact of the stress. So phobic anxiety is amenable in that it is a diagnosis that occurs early on in life and continues, unlike certain forms of depression that are episodic and harder to sort of get a handle on. We also looked at PTSD in collaboration with Kirsten Conan, who was my friend and colleague who got me involved with LaCoy. And we looked at the impact not only on civilian women who were sufferers of violence, but also military folks. Another interesting work that I'm doing right now with colleagues in the South, as well as my colleagues here at BU, is racism. Every day. So how do you capture racism among African Americans? So we have a couple of papers where we show that everyday racism was associated with shorter telomere length among women who reported not discussing those experiences with others. In other words, women who didn't vocalize or didn't share their experience of racism had a disproportionately lower, shorter telomere length than those who did. And again, here is a very, very simplistic mechanism model that we put together to show how positive exposures versus negative exposures impact telomere length. And again, it all runs through oxidative stress and inflammation, but there are some genetics to lead to early aging and chronic disease. In addition to, I've focused exclusively on psychosocial stress in this talk, but I could tell you that my MO is that I tend to want to leave on a more optimistic note. So we know that there are some positive lifestyle factors that one can adapt to not lengthen your telomeres. I neglected to say that once you shorten your telomere length, it's not reversible. Okay, you can't turn on telomerase, for instance, but you can adapt certain lifestyles that actually buffer or prevent accelerated shortening. And so I'd like to thank my lab. The lab folks who are specifically involved with these studies are indicated above. And with that, I thank you very much.
Hi, so I'm also reporting live from Boston. So good morning from the side, and afternoon and evening from wherever you are. So these are my affiliations down there, and the topic has already been introduced. So in talking about epigenetics, I think there are two things to sort of like think about. Obviously, one component is as genetics. So in this figure here, or in sort of here, I'm sort of thinking about the question, how much of the phenotype that you're observing or the ones that you are interested in is attributable to genetics, and how much of it is attributable to the environment? And the answer really is that it depends. So I'm showing here, and also it depends on the heritability of the phenotype you're looking at. So this figure here shows different psychiatric illnesses down there and their heritability rates. So in the pink, it's showing heritability rate estimates from twin studies. So in the twin study model, what we're looking at is what is the likelihood of one twin having, or in this instance, a mental illness when the other twin has it as well. So it's very clear then that specifically for monozygotic twins, that the likelihoods are very high, also actually depending on the phenotype. So let's say, for instance, in bipolar disorder here, the observed heritability is over 80%. So when we're looking at SNP heritability, that is what is shown here in the blue shading, and SNP heritability is essentially what we've been able to observe in the genetic studies that we have conducted so far, is that there's quite a big difference between what we SNP heritability and twin heritability. But of course, then there's this other portion here to be able to explain, though, to the full extent, the phenotype, this other portion here that is attributable then to non-genetic and environmental factors. So the phenotype we're seeing is sort of a balance or interplay between genetics and environment. So as already mentioned in the introduction, my research has been on schizophrenia for the most part. So that's what I'll speak about in the next couple of slides. So there's a study ongoing in South Africa now called Genetics of Schizophrenia in the South African Xhosa population, called SAX. And in this study, participants were recruited from the Western Cape and Eastern Cape provinces in South Africa. And these are people who self-identified as Xhosa. There's a lot of analysis done here, and I'm only showing sort of one figure from the main publication back in 2020. So this figure here is showing that, so there's two different studies. In a different study, they looked at sort of gene dysregulation from people who had passed on and had mental illnesses. So they're showing that some genes are sort of upregulated and downregulated, so meaning not functioning as one would expect. So when looking at those genes in the study population, we're seeing that in the red bars are the cases and the blues are the controls in the SAX study. So we're seeing that amongst the genes that are dysregulated in schizophrenia, there was a higher rate of sort of those genes that were downregulated in this population, as well as those were downregulated for autism as well as upregulated in bipolar. So really sort of kind of getting to grips with understanding how the disorder is caused. So again, from previous studies, this dysregulation, we're seeing sort of a similar pattern of dysregulation in participants in the SAX study. When looking at environmental factors, and very quickly, we kind of the papers that came out from this study that showed that people who had schizophrenia in SAX had also been exposed to high levels of physical and emotional abuse, for example, and had sort of overall higher rates of trauma. And looking at substance use, we see sort of higher rates of substance use disorder in people with schizophrenia. So when sort of connecting the genetics and the environment, people who have sort of a genetic burden for schizophrenia also had very high levels of substance use disorders. So there's been a question about, is it the genetics that causes people to have sort of to use substances, and sometimes it's like a coping mechanism, but also the reverse can be, the relationship can be sort of in the other way, where is it the substance use that sort of causes people to, develop these mental illnesses? And this is kind of a question that we're grappling with. So moving on to another study called the Drakenstein Child Health Study, also in South Africa, where women are recruited into the study during their second trimester of pregnancy. So the moms are recruited, and then them and their children are followed up after birth. I think the kids are now about 10 years old. So this study is really looking to see what are the psychosocial exposures and exposure to traumas, in what ways do they influence or do have an impact in the children. So this is sort of more looking for intergenerational effects. So there have been some publications that come out from this, and essentially showing that in general, these women have been exposed to high levels of trauma. And as far as what effects this has on their children, it has been shown that women exposed to trauma, the effect in their children is that the children have sort of reduced head circumference. Intimate partner violence has also been reported at high rates, and the outcome in their children is sort of this low infant birth rate. For post-traumatic stress disorder, there is adverse neurodevelopmental outcomes specifically at 6 weeks. And what this kind of looks like is children aren't able, for example, to perform tasks like, holding the bottle while being fed, and that sort of thing. So thinking about the, how this is happening, how this trauma event that has happened in women are affecting their children. I think the real answer, and perhaps what we're talking about here today, is that the DNA keeps a record, as Professor Debo has spoken about. So one of these mechanisms is sort of DNA methylation. So the DNA itself isn't necessarily changed, but there are changes on the DNA itself that sort of regulates, how genes are expressed, whether they are sort of downregulated or upregulated. And this is really the cells or the body's way of trying to maintain a sense of like regular operation. So in DCHS, it's been shown that there's a difference in sort of gene expression between the moms who have had these traumatic experiences compared to those, and there's been a correlation as well with, for instance, cognition in their infants. So the studies that I've spoken about, the two studies that I mentioned, are at this point sort of in their infancy, in that the sample sizes that we are conducting these studies on are not as massive or as big as what's required for the genetic analysis that we do on the one hand. For example, with genome-wide association studies, I think studies that have been done in specifically European ancestry populations are now in about the number of cases is about 60,000 or so. So the SAX study, the analysis that I showed was in just under 2,000 individuals. And for Drakenstein, this is just under 1,000 individuals. So I think to move closer to the truth, and what I mean by the truth is, understanding disease mechanism, also like taking into account all the risk factors to be able to provide better care to the people who are affected by these illnesses. So I'm just kind of have sort of 3 small bullet points here. One is that we need to have large-scale epidemiological studies in African populations. Again, what is the prevalence? For instance, when I was doing my PhD, a lot of the prevalence studies, or a lot of the studies that report on prevalence were done in the global north, and there isn't a real sort of account of prevalences in other nations. It's been said that, it's expected that they'd be similar, but we don't know that for sure. Also because we know that there are sort of different levels of reporting of these illnesses because of, how they understood culturally. So that could hinder sort of the prevalence statistics. I've already spoken about the lack of genetic studies in African populations. So Eurogap, for example, is a study that I'm doing my postdoctoral fellowship on right now. And we're recruiting individuals from Ethiopia, Uganda, Kenya, and South Africa. And we're aiming to recruit 40,000 individuals in total. So this is, again, working towards, having a representation of African diversity on that scale. And lastly, we need to be able to adequately model environmental factors, because there are so many. And when we're kind of moving towards building up polygenic risk score, that's been quite a hot topic recently, where we're looking at a person's genetic burden versus the environmental risks, and putting that together to say your likelihood of developing this sort of disorder or this kind of phenotype is this, based on the summing up of all those risk factors. So I wanted to touch a little bit on sort of the considerations that would go into these sorts of analysis. One of them is maybe also very important, cultural perspectives. So in the SAX study, what came to light was that there are sort of cultural differences, or differences in general, about understanding of the schizophrenia in general. And for instance, like, without even talking too much about it, like, there isn't even an equivalent or a word in Xhosa that means schizophrenia. And there are sort of cultural beliefs. So in people who have schizophrenia, during the interview process, express that they believe, in the way that they understand their illness, is that they were bewitched and somebody is out to get them. So if you're thinking about sort of that cultural framework, maybe you could be thinking about if we incorporated that sort of cultural perspective in the way that we diagnose people, because if you incorporated that into sort of the DSM, for example, would that sort of have a better outcome for patients? Would there be better health-seeking behavior? There are other sort of a consideration, which could be socio-demographic, like what are the environmental risk factors that could be present in these populations that are not, for example, like exposure to like HIV infection and TB and so forth. So so they are sort of these three, I've kind of highlighted these 3 ongoing studies. So SAX is in a second round of funding, and they're recruiting trios. So this is more, again, looking at sort of the intergenerational effects of genetics and environment. So getting towards the marrying of genetics and environment in epigenetics. So thank you very much.
And thank you, Dr. Machara and Professor De Vivo. Very exciting presentations. And so both of you covered very different mechanisms that actually can end up silencing the gene, for example. So the idea of the epigenetics, of course, is that the genetic code is what is sort of, could be the same that is transmitted, but the gene itself how is it expressed is modified by many factors. So it's akin to reading a book. Like you have the genetic, the code is there. You might read a certain paragraph or a certain passage, but if that page is kind of stuck or something is blocking you from getting to that page, then you might never be able to read that paragraph. And this is what epigenetic does. It kind of silences certain portions of the gene so that they are not expressed. And so Dr. DeVivo talked about the telomeres as one mechanism that actually can either put a dimmer switch on the gene or silence the gene. And then Dr. Majara talked about methylation, which is another mechanism that can silence the gene or prevent access to the gene for expression. So here you're talking about not that the gene has changed, it is how you're reading the gene that is being changed by what is sitting on or changing access to the gene. So, like I remember when I was going to the university, one of my professors was talking about this. He said that a reporter had come to me one day and said, what controls the expression? Why are you the way you are? Is it a product of your genes? Are you entirely dictated by your genes? Or is it the environment that's making you the way you are? And so he thought for a minute and he says, young man, he says, it's like asking what contributes to an area of a rectangle. Is it the length or the width? It is really both. And so that is the idea that the information that is being transmitted through the genes is one component, but in itself is not enough because the gene itself is not fully expressed or selectively expressed. And as life events can modulate how the genes are being expressed. And not only that, Michael Meany from Canada has shown that this information can jump generations so that if you have a certain methylation pattern on your gene because of early childhood trauma, that you will then pass on that information, gene markers, to the next generation. So that's how you can explain transgenerational trauma, transmission of trauma, for example. So keeping that in mind, I was going to ask both of you a question, and that is, if I bring a context more sensitive to the Asian continent, the Asian context, there you see that the Asians have been found to be about 6 times more likely to develop type 2 diabetes. So we don't know quite exactly know what the cause is. And certain hypotheses put forth saying that maybe the Indian continent had faced so many famines, like up to 31 major famines where people died of starvation. And that has programmed transmitted information to make the bodies that are adapted to it so that you're not developing lean muscles that actually burn a lot of energy, and you learn to store fat as a protective mechanism. And so maybe that contributes to why this continent has so much expression of type 2 diabetes. So my question to both of you, and feel free to comment on it, is to say that is this preponderance of type 2 diabetes in Asia, is it due to genes or is it due to the environment, or is it something to do with your lifestyle? So how would you know whether the effect is sort of epigenetic in phenomenon, so that your genes are not changed, but how they're expressed is being changed, and that has been, the stage has been set up by what happened in the past. So I'll open it up and to see what you think about that.
Start, and then Dr. Majara can speak. So we know that there are predisposing variants to type 2 diabetes, and we know that there are certain groups that have a higher risk of type 2 diabetes compared to others in the United States. There's extensive study on the Pima Indian group that have a disproportionately high prevalence of diabetes. And the answer is exactly what Dr. Majara said before. It's a little bit of both. especially in these diseases that do not have a Mendelian inheritance pattern, right? Where Mendelian inheritance suggests one gene, one disease, where there's many genes and many differences across the genes, many variants across the genes together in conjunction with lifestyle factors, absolutely lifestyle factors. And I would argue that most of the world was under famine for eons. And so I would argue that it's not exclusive to one group or the other. However, and I don't argue with the historical evolution that you mentioned, because it's all, yes, it's pretty very much consistent. I would argue that the increase in the availability of very bad food at a very low cost puts people at risk that already may have underlying predisposition to something like diabetes type 2. And the evidence for that is indisputable, unequivocal. It's in many papers. So it is a combination of, yes, your genetics and your environment, but particularly a very poor environment right now with respect to food. And I would now go to Dr. Majari.
Thank you, Prof. Yeah, I think you have said it all. I'll just add on just a little bit. Sort of expanding on, you mentioned the Mendelian sort of inheritance pattern, where one gene, you will definitely see the phenotype, or in this instance, definitely develop diabetes, but that's not the case, because diabetes is quite polygenic. So many different genes, each one contributing a very, very, very small, minute risk. So that's to say, if you've got the genetic propensity, it's not guaranteed that you will develop diabetes. And then on the other end is, okay, yes, you have a propensity, and the heritability rate of diabetes is what, 40%. So let's say you have a 40% genetic burden, or 40% risk for developing diabetes because of your genes. That means that there's still about 60% that is attributable to other factors, including the environment. So Professor DeVivo has spoken about access to food and nutrition, and that how that could be sort of contributing to the high prevalence of diabetes. And so there's that access, but there's also sort of lifestyle choices that people make. So what that means is then, like, if you've only got a 40% propensity genetically, then all the other things obviously taken into account, the risk of diabetes then could be mitigated, because if you didn't eat unhealthily, if you did exercise, practiced mind body kind of therapy, like the effects could be mitigated. So it's that interplay, and a lot of it is to kind of do with and I'm not taking away the genetic risk, but I'm just sort of trying also to say that a lot, like, there's also things that an individual can do to mitigate getting diabetes, I guess, in those instances.
And I might add that the genome genes are, for the most part, unmodifiable, right? But the epigenome is modifiable, and that's what we're learning today. And that's where we as a population or people can take some control over that. We can't control for what we've inherited at the gene level, but we can do what we can to keep our epigenome as healthy as possible by making the lifestyle choices. And exercising is one way, and eating healthy is another way. And I didn't mean to undercut anyone, but I wanted to anyway share that. Yeah. So this is mixing a couple of things. So I don't think I said that there's a life expectancy high in one versus the other. And I could see why that would be, someone would go to that conclusion. So what I was mentioning is that what we know, just some of the telomere dynamics that we've learned. And in that, it's the paper I've included in the lecture is adaptation evolutionarily. So we know that hunters and gatherers have longer telomeres compared to African Americans, compared to Caucasian of European descent, because of this is not there's no disease related to it. It's just a fact, in the same way that young people have longer telomeres than older people because there is accelerated shrinking. But I could see how someone could mix the two up. And again, the paper refers to evolutionary adaptation. This is one of the reasons, and why African Americans would be in between Africans, hunters and gatherers, versus European, Caucasian of European descent, is because African Americans have on the order of anywhere from 20 to 80% of mixed lineage with European descent. There's admixture. So it's not a question of living longer. There was no, I'm sorry if there was any confusion to that. Those mortality studies were based on, in that case, the Danish paper is, the Danes are of European descent, but I don't know, I'm sure if someone did work on papers of other ethnicities, we might have a broader picture. So that is just, we only can talk about the data that we have at hand. And Dr. Majora, as you said very well, is that a lot of the studies right now are based on participants that of European descent, and so we are expanding, broadening, and we're finding out more and more when we include folks of different ethnicities.
So I think that we're certainly out of time, and wanted to thank our guests for sharing of their time and their knowledge with us. Thank you, participants, for participating in this webinar. Hope you gained a lot of insight into the gene debate to understand our human behavior. And then finally, I wanted to say a thank you to Candy and her team for managing and putting together this webinar in such a professional way. So I'm hoping that we all had a great time and it was a great learning experience. And thank you very much all. Bye-bye.